Hello, welcome back to the discussion on horizontal gene transfer in bacteria. Today is the part 2, where we will be discussing on the bacterial transformation. Transformation is a process of genetic recombination in which a DNA fragment from a dead, degraded bacterium enters a competent recipient bacterium, and it is exchanged for a piece of the recipient's DNA. It is an energy-mediated process. Transformation may alter the phenotype of recipient cell. Here we can see the isolated DNA. They are entering in a bacterial cell, and after crossing over, this incoming DNA is incorporated within the host chromosome. Transformation can be accomplished by two ways: transformation with DNA fragments and transformation with a plasmid. In the first case, DNA fragment can be inserted within the host chromosome through non-reciprocal recombination, or it may be degraded if recombination doesn't occur. It is called the unsuccessful transformation. The DNA plasmid as such can be entered within the bacterial cell. Now the question comes, why is transformation required for bacteria? The scientists found three answers. The first answer is the bacteria use this phenomenon to acquire resistance from the foreign DNA, to acquire resistance against antimicrobial compounds. Second answer is for gene repair, that is to compensate the loss of one gene or deactivated gene can be replaced by gene coming from incoming DNA. The third answer is it can be used just as food. Let us have a quick look on the history of the discovery of this phenomenon. In the year 1928, Frederick Griffith, he discovered the phenomenon of bacterial transformation. He was working with Streptococcus pneumoniae or pneumococcus. He took two types of strains. One is rough, non-virulent, that is strain 2R. Another is smooth, virulent strain 3S. In an experiment, he took rough strain and smooth strain. He injected rough strain in a mouse, and as it was non-virulent, the mouse didn't die, and he could isolate the R colonies from the tissue of the mouse. Then he injected smooth strain in the mouse and found the mouse was dead, and he found the S colonies from the tissue of the dead mouse. Now he heat-killed the S strain, and now he injected this heat-killed strain in the mouse, and the mouse didn't die, and there was no S colonies in the plate. Now what he did, he mixed the non-virulent strain along with the heat-killed virulent strain and injected it into the mouse. It was expected that as there was no virulence, the mouse may be alive, but the mouse was dead, and he found both the R and S colonies from the tissue of the dead mouse. From this phenomenon, he suggested that perhaps some change has been taking place, and for this change, the non-virulent strain was converted to virulent strain, and he termed it as a transformation. There are two types of transformation: natural transformation and artificial transformation. Most types of bacterial cells cannot take up DNA efficiently unless they are exposed to special type of chemical or electrical treatment. But some bacteria, about 40, are naturally transformable. They are Bacillus subtilis, Streptococcus pneumoniae, and Helicobacter pylori. Naturally competent bacteria carry sets of genes that provide the protein machinery to bring DNA across the cell membranes. The transport of the exogenous DNA into the cells may require proteins that are involved in the assembly of type 4 pili, type 2 secretion system, and DNA translocase complex at the cytoplasmic membrane. Now we are coming to the competence in gram-positive bacteria. Whenever the bacteria can sense the presence of isolated DNA in the medium, the bacterium releases competence factor in the medium. When the titer of the competence factor reaches or crosses the threshold limit, it will bind with the COMD protein present on the surface of the bacterial cell wall. COMD protein then serially and gradually activates the protein, the genes present on the COM operon, which will ultimately form the gateway through which the incoming DNA can enter into the host cell. This complex is called the translocasome. Now, the protein involved in the transformation of gram-positive bacteria is a product of COM gene. This gene is organized in several operons. The product of COM-A and COM-K are regulating competence, whereas COM-E, COM-F, and COM-G encode for structural proteins for uptake of DNA. Now the first gene of COM-E operon, that is COM-EA, encodes the protein that directly binds extracellular double-stranded DNA. Whereas the COM-FA gene, the first gene of COM-F operon, is actually an ATPase that helps in the translocation of DNA into the cell. COM-G gene of COM-G operon encodes for a protein that forms pseudophilus, the channel through which the DNA enters. Nuc-A gene encodes nuclease enzyme that cuts extracellular double-stranded DNA to single-stranded DNA. Single-strand bind protein and REC-A product helps in combination of incoming DNA with the DNA of recipient bacteria. The competence in gram-negative bacteria, a variety of gram-negative bacteria are capable of competence. Some examples are Acinetobacter, Helicobacter, Neisseria, etc. They utilize two different pathways for uptaking DNA. One is PSTC transformation pathway, another is type 4 secretion related pathway. In PSTC, P stands for pilus formation, S for secretion, T for twitching motility, and C for competence. Here, the entry and insertion of the DNA strand occurs through competence pilus and ATPase, then DNA pooling and compaction at outer membrane, inner membrane translocation, and finally single-stranded DNA recombination. Type 4 secretion related pathway, which is found in Helicobacter pylori, here the DNA is translocated through cell wall and membrane with the help of protein similar to agrobacterium for conjugation of TI plasmid. Type 4 system function transfers of DNA in two ways, moving in and out of the cell. Genes located close together are often transferred as a unit to recipient cell. It's called the co-transformation. And similarly, the genes that are far away are less likely to be transferred together. These are all about the natural transformation. Then most of the bacteria are not naturally transformable. These bacteria can be made competent by certain chemical treatment or by strong electrical shock. First one is a calcium treatment. Treatment with calcium ion make some bacteria like E. coli, Salmonella, Pseudomonas competent. When the calcium ion increases the permeability of the cell membrane, and cell treated with calcium along with heat shock can take up both single-stranded DNA as well as double-stranded DNA, no matter circular or linear. The next procedure is electroporation, where a strong electric shock is applied in the bacterial culture mixed with the naked DNA. The recipient bacterium is prevented from osmotic shock. The strong electric field creates artificial pore of water lined by phospholipid head group through which the DNA can pass, and it is the artificial hydrophilic pore. Now, what are the applications of transformation? First one, it is applied in the recombinant DNA technology to make multiple copies of DNA in DNA cloning, to make huge amounts of specific human proteins like human insulin used to treat diabetes 1, to genetically modify a bacterium or other cell. So this is all about the bacterial transformation. Next part will be coming soon. Till then, thank you.